Hi, I'm Dr. Lauren Launen, and this is the first of a multi-part online lecture aimed to introduce you to the microbiology and a little bit the genomics of a research project conducted in my lab where we aim to characterize Vibrio vulnificus that are isolated from the Great Bay estuary. The first part of this lecture is to introduce you to some basics of microbiology. So I'm going to introduce you to microbes, give you an introduction to Vibrio vulnificus, talk about why studying Vibrio vulnificus in New Hampshire is important, and give you our project goals. And in the second lecture, I will give you an overview of the project. This first part will simply be an introduction to microbes with a little bit of an emphasis on Vibrio vulnificus. So a microbe is a single-celled microscopic organism or virus. All microbes are able to live as independent entities. So unlike you or I, which require many thousands of cells to live, to perform our metabolisms, and to reproduce, each microbe can accomplish all of the goals of life, which are listed here, only as a single cell. A pathogen is a microbe that can infect and cause disease. That's a small group of microbes. Most microbes are either harmless or in fact can perform useful functions both for the human body or other hosts and also simply for the planet. But a handful of the microbes have an evil side to them, and we call those pathogens, microbes that can infect and cause disease. So all pathogens have to have phenotypic characteristics that allow them to infect and cause disease, and we call those characteristics virulence factors. So a virulence factor is a physiological ability that enables pathogenicity, and each virulence factor has to be conveyed by specific genes, or perhaps more accurately, particular alleles for certain disease. So in that equation down there, D stands for D, N stands for the number of microbes, V stands for virulence factors, and the denominator R stands for resistance. The amount of disease that you get, or D, is going to be a function of the virulence factors that the microbe infecting you is associated with. So in microbiology, a central question that drives many of us, and it certainly drives me, is the question, what makes a pathogen a pathogen? Or in other words, why are some strains of the same species pathogenic while others are harmless or even helpful? And this cartoon is depicting it, that relationship there. We've got two identical looking microbes. These are eukaryotic microbes, you'll note. And one of them saying, "You make me sick." The other one is obviously a pathogenic version of this species. So just to go back to the idea of virulence factors is in fact part of the answer to that question. So particular strains within a species can have alleles that encode harmful proteins that can convey harmful abilities. And in Vibrio vulnificus, the organism that you're studying in your project and that we focus on the lab group, there are a set of what are called putative or potential virulence factors. And some of them are listed here on the left. So on the left are the names of the genes, and on the right is what those genes encode, or the phenotype that those genes encode. So for example, there's a gene that encodes an epimerase protein and a gene called WPB, as well as a set of others. And in combination, when those genes are expressed, it gives Vibrio vulnificus the ability to make capsular polysaccharide, or in other words, to make a capsule. And this is a classic virulence factor you were introduced to when you learned the story of Fred Griffiths and read about his work on Streptococcus pneumoniae. Vibrio vulnificus is no exception. Having a good capsule is definitely a virulence factor. The VVHA gene and the RTXA1 gene both encode toxins, protein toxins. A hemolysin toxin allows Vibrio vulnificus to break red blood cells. This SMART AVV toxin is a cytolysin toxin, and it allows Vibrio vulnificus to damage host cells. VVE encodes something called an elastolytic protease, so that's a protein that lets you break down certain types of protein in the host, better facilitating infection. SOD-A encodes a protein called superoxide dismutase, which allows Vibrio vulnificus to fight back against immune system attack. Skipping down a little bit, pill A and pill D allow the microbe to make proteins that allow it to build what are called pili, or attachment structures. These allow Vibrio vulnificus to attach to the host and also to sometimes inject it with the aforementioned proteins. So I won't go through all of these factors, but I want you to be aware that possessing certain alleles of these genes will allow Vibrio vulnificus strains to be virulent, while others which possess different alleles will not be able to show virulence. So going back to some fundamentals of microbiology, I want to make a couple of very, very important points. These are single-celled creatures, and they are very tiny, generally speaking, when compared to eukaryotic cells. A quality of tiny cells that you should remember from your first-year biology classes is that tiny cells can replicate very quickly. So if you can replicate very quickly, that means that you can evolve very quickly, because all of that replication will be accompanied by a certain amount of mutation. And also, as you know, prokaryotic cells, to which Vibrio vulnificus belongs, are capable of genetic change through the 3 horizontal gene transfer mechanisms: transformation, transduction, and conjugation. So that means that they're able to change their genetic nature relatively quickly, and this favors very rapid evolution. When you can evolve rapidly and you can replicate rapidly, which results in large cell populations, it means that you can see very, very extensive diversity within a species. And so this is a complicating factor in microbiology, but also one of the things that makes it fascinating. If you look at a single population of the same species or even of the same strain, you can see incredible amounts of genetic variation or diversity within that population. This is one of the reasons why we can see single species possessing multiple strains, some of which are pathogenic and some of which are non-pathogenic. So with that, I'm going to ask you to go to the next part of the video.